Thank you for staying all day long and now listening to me. So as I was introduced, I'm a virologist, I'm a veterinarian, so I work in a laboratory, so I'm a practical example on how the laboratory, the field veterinarian, and the GIS people can interact to solve a problem or to prevent a problem. Both my cases will deal with viral diseases, namely two important viral diseases, viral hemorrhagic septicemia, in short VHS, and infectious hematopoietic necrosis, in short IHN. These two viruses are really important for rainbow trout production because they cause losses in the aquaculture, and also according to the European legislation, they are notifiable disease. Actually, they have been listed according to category C. That means that the disease can be voluntarily eradicated on a voluntary basis. So a disease and a territory can decide and choose if being free or remain infected or move from a category to another. So let's start with the first one example. For this example, we are here in the northern part of Italy. This is an autonomous province called Trento province, and it is located, as you can see, in the northeast part of Italy on a mountain region. It's a mountain region in the Alps, and this region is really important for Italian aquaculture. Rainbow trout production in Italy is really important, and for this region is the most important aquaculture production due to the orographical conformation of the geographical conformation of the territory. It is rich in fresh water, in spring water, so a lot of rainbow trout are farmed here. Notably, the first rainbow trout in Italy was introduced here in this region back in 1861. So this region has a long history and experience in in farming rainbow trout. And during this long period, they tried to eradicate the disease, and actually they succeed in 80% of the territory, and they have 16 zones here, completely free, officially free, and recognized at the European level as officially free of the diseases. And we can also see the number, these are the number of the free farm in Italy. Of course, the majority of farms are located on the mountains, here in the Alps. And here we have the highest number of officially free rainbow trout farms in Italy. Unfortunately, still they have some infected farms in the territory, but the effort of the province is to control even this one, and so they continuously monitor all the trout farms present on the territory. So this case, so this is an enlargement of the region here, thanks to the GIS, of course. It is a map drawn by the GIS. You can see how many trout farms exist in this territory. The blue dots correspond to the establishment of aquaculture farm of rainbow trout and other salmonids. And the red square correspond to the put and take fishery. Tourism is another important activity for this region, and angling is a very popular sport, especially during summertime, due to the beautiful landscape and to the climate the fishermen can enjoy here. So this is the first problem we encountered, so a mixture between recreational activity and industrial activity. So the case I'm going to present you took place here. Can you see the dotted line blue here? This is a free zone. It is called Val di Sole, meaning sunny valley, an evocative name of the place, so a beautiful place, starting from following the stream of a river called Noce. In this slide, okay, this is again another map. Closer, here we have the spring of the river, And here we have the barrier at the end of the zone. You know, according to European legislation, to identify a zone, we have to identify a precise geographical location, so from the spring to a barrier capable to prevent the movement of aquatic and wild animals, aquatic animal in general, so to control the disease. So this area is epidemiologically separated from by the dam here. After the lake, here is a lake called Santa Giustina. So we had a problem. We found a positive result in my laboratory. It was an IHN positive results in a farm, actually a put and take fishery, so a small recreational farm here. And as you can see, it is exactly in the middle of the free zone. In this map also, you can observe there are other farms, and we will see in detail later on. So this is a picture of the put and take fishery. Actually, it is a real tiny lake, but the problem is that the lake is connected to the river Noce. So the inlet water was here and the outlet water was here. So an outbreak here actually is a risk through water connection to the other farms. Immediately, an epidemiological investigation started, a standard epidemiological investigation with the colleagues from the field going there, collecting other samples, checking how the fish behave. And actually, they realized that there were just a few specimens, trout in this put and take fishery, no clinical sign at all, no mortality, nothing happened. So they investigated more and they realized that there was an angling competition some weeks before with an unauthorized movement, and we actually couldn't trace the origin of this movement of the fish introduced in this farm. So this was probably the event producing the outbreak. Another interesting point I have to stress that in a free zone, a put and take fishery, if connected to the water, is a risk factor. And so also, this small activity undergoes surveillance, and it was thanks to the surveillance that we could identify the positive result in the laboratory. So, again, the importance of surveillance, everything inside a zone. So, as I told you, an unauthorized movement of fish was identified. We characterized genetically the sequence of the virus isolated, and the sequence corresponds to the red one. The other are Italian strain, the yellow and the green, recently isolated from the infected farm inside the province, but also outside. So it's not exactly the same virus, but very similar. So probably the introduction is not something really far from our realities, came from some infected zone. I remember you that the rest of Italy is not free, so potentially infected. So actually we couldn't trace the origin of the fish introduced in the lake, and so could come from anywhere else around, in the territory around the free zone. What we decide, considering the characteristic of the outbreaks, is not to withdraw the free status of the entire zone. According to the European legislation, if in a zone we have an outbreak, the free status is lost immediately. But in this case, there were no clinical signs, few fish, and then we will see also the characteristic of the other farm connected that are really far and hosting also Salmoni species, not rainbow trout, that is the most susceptible species, but also local Salmoni species that are a little bit more resistant to the disease. So together with the local veterinary authorities, we decided to take advantage of the Article 42 of the Animal Health Law legislation, which allow the suspension of a free zone, investigating what is going on before withdrawing the free status. So this was the choice we did. Of course, we have to justify this solution, and we plan increased surveillance activity in the establishment connected, and also extra sampling in the wild. So this is thanks to the GIS, and this is how GIS really can help during an event like this. The data small, but actually we've drawn immediately a list of all the establishments insisting in the zone. We could map those upstream, so actually we forget a little bit about the one upstream, sorry. We listed those downstream, and also it was really useful to calculate the distance by water connection from each establishment connected to the water, because in some case, as you can see, some establishments were independent. So if they take the water from other sources, not the river, they are not connected to the establishment. And as you can see, as I told you, there are large distance from a farm to the other. So the most close was 11 kilometers downstream, and the farthest was 21 kilometers downstream. So long distance. There is a EFSA opinion where in a river, a dilution of the virus happened after 5 kilometers of water stream. So we are more than double of the distance. So we could really, in an afternoon, in a few hours, decide which farm sampling, when, and how much. So that was what we did, and we decided to sample all this farm, and also to collect wild fish in order to increase the guarantee of surveillance in the territory. At the same time, the outbreaks was eradicated, so according to the European legislation, 689/2020, so it was empty, it was the lake empty, it was cleaned, disinfected, followed for 6 weeks, and then the water and the fish was reintroduced in the farm. At the end of the story, just to make a long story short, we perform sampling, everything tested negative, even the wild fish, even the sample in the farm, and even we collected fish after the reintegration of the water inside the farm, according to European legislation, 3 months after introduction, but before 12 months after the introduction. So we have this window where we could sample fish, a lot of fish, 150 fish actually, and they tested negative. So thanks to the GIS, we could rapidly solve, but also thanks to the classical epidemiological, to the rapid field inspection. So it was actually a teamwork, but we could really control an outbreak in a relatively short time, and the most important thing, without losing the category. So the other farm could continue to work, even with some restriction in movement, but they were obliged to clean, to empty also their farm, because no positive result beside the one in the lake was found. So this was a happy end for us, and I think also for all the authorities and the farmer involved in the outbreak. And this is the second case study. So for the second case study, we move southern in the central of Italy. In this case, we did not experience a problem, but actually we had something to solve. So in this way, the GIS was used as a preventive way to analyze data. We are here across these two regions, Laser. This is Umbria region, and this is Marche. And on the border, so across the two regions, it is located the Monti Sibillini National Park. This is a very beautiful national park, very big, because it's 70,000 hectare extension, so very large. It is located again on mountains, a different mountain chain. In this case is Appennino Centrale mountain chain, and the average height on the level of the sea is between 360 to 2,400 meters, so quite a hill and mountains territory. And the Monti Sibillini National Park was born to protect biodiversity, local biodiversity, both terrestrial but also aquatic, because it's another area rich in water. And in particular, there is this project that is an European project called Life Stream that aims to protect a local autochthonous trout. This is a Mediterranean trout, Salmo gigi. That is a very interesting species locally, and also protected because it's endangered. And among the aim of this project, there was the eradication of the alloctonous species, the reintroduction of these autochthonous species, and also, if possible, to make the entire area of the park free of disease. This was the aim. At the end, I will show you reduced our goal, but it was indeed a good result. So, again, GIS map. These are the park. The boundaries are in red, and this is the river reticulus, so the hydrobiological network of this area, and as you can see, is quite complex. So this map shows 6 colored areas. Actually, we started with 11 areas possible, if possible, in feasibility study, we identify 11 areas. Then we reduced to 9 and finally to 6. So these are the 6 zones that at the end were not approved, but we subjected to European Community the eradication plan. So why, and these are the names, as you can see, 5 are inside the boundaries of the national park, 1 is outside. Because, why we choose this one, again, to make a long story short, because it's a 2-year working activity, this 10-minute talk, it was a huge amount of work. Because we also couple, thanks to the grant of the project, ecological and genetical study. So river by river, they actually calculated the amount of trout population existing present in river by river, which species they belong to. As you can see, there is the Mediterranean, so the local one. There was the Atlantic, that is actually an alloctonous species and is a main competitor for the local trout. And then we have a number of hybrids, because the main risk for the local strain is the hybridization with the Atlantic trout. So according to this data, so the maximum number of fish found, and also they calculated the maximum number of fish we could collect to perform the test, the laboratory test. We have to kill some fish to test them in the laboratory, so without impacting, of course, in the population. And also according to the genetics present in the river. So depending on which river, we select the area of interest to eradicate the disease. And these are just 2 examples. I don't want to bore you, I'm not presenting area by area, but just 2 examples. This area is called Altonera, that is actually the name of the river from the spring through the barrier. Then we also inspected visually each barrier to understand if the barriers can really avoid the movement of wild fish and could act as a real barrier. In this case was a fall. So we measure also the height of the fall to be sure that trout could not jump this fall. Where present, we also mapped the establishment, so farm, because there are also farms in this area. Of course, we mapped mainly those connected directly to the water stream, because this can be also used as sampling point for checking the animals, as this was the case. But in some other area, like this, a beautiful area indeed, the barrier was a dam, so easily to be recognized as a barrier. But here we couldn't find any establishment where a fish could be sampled. So thanks again to the GIS and also to local inspector going and looking to where it's feasible, we identify some tracts that were actually mapped geographically where wild fish could be sampled. Again, to be tested then in the laboratory. So these are some pictures of electrofishing activity in the river. Of course, we selected and we choose for testing the rainbow trout or the Atlantic trout is present, so the alloctonous species in order to eradicate them. But also we could find beautiful specimens of the Mediterranean trout, and in this case we collected, where feasible, non-invasive sample like ovarian fluid or reproductive fluid in general, so we didn't kill the fish actually. These are really nice fish. They fished. And so they are happy now because we presented the eradication plan to the European Community. They were approved. And also we finished the first year of sampling. Some of the eradication plan last 2 years, some other 4 years, depending on the maximum number of fish we could collect. But we are optimistic that by in 2-4 years, depending, we will reach the free status in all 6 zones we identify. And these are just 2 practical examples of how GIS can integrate with laboratory activities, so my part of work, and also the field veterinary officer, the official veterinary officer activity, that it's really important that all these players work together to reach such a result. Thank you.